Okay, this is the third video for the week. This is the bacteria of the skin. This is a lab that actually goes over a 3-week period. So with all of our holidays, it's going to be like 4 weeks. So anyway, for this one, it's pretty simple. You're going to have a red plate. This one's kind of faded in color, but it's called mannitol salt agar, and it's a specific type of agar that, just like it sounds, it has mannitol, which is a sugar, and salt, which is salt. So the bacteria that grow on your skin can grow in a very high salt environment because you have salt on your skin. And the mannitol is to see if it can ferment that sugar. So specific bacteria that live on the skin, like Staph aureus and things like that, will be able to ferment the mannitol as well as live on this plate because of the salt. A lot of bacteria, this is a bacteria that only allows salt-tolerant or halophiles to grow. So other bacteria that can't withstand that osmotic pressure of the salt won't grow on it. Okay, so it's pretty selective and it's differential, so it's kind of a good medium to use for both. So what you're going to do for this is, like I said, it's pretty simple. And each person will have their own plate. Even though you're working in groups, everybody does their own. I want to get as much variety as possible. And select a part of your body to get the swab from. I suggest that you use a part of the body that's maybe a little bit sweatier than others. Get as creative as you want without being too creative. I've had people do their cleavage. I've had people do different places. So, well, not too bad. So just, you want to get a place where you're going to try to get some bacteria that's going to grow, okay? By the time you're done with this lab, you'll be able to identify down to the species level what type of bacteria is your normal microbiota that you have growing on your skin, okay? And so what you do is, once you've selected the site, you do between your toes, behind your ear, your nose, things like that. If you do just your hands, they're usually pretty dry and you don't have a lot growing on them. So what you're going to do is take one of the sterile swabs, and then you're going to use some sterile water. And it's just enough water to moisten it. Okay, and it might actually be saline that we're going to use. Yeah, it is saline, so it'll have salt. Okay, so it'll be sterile saline. So anyway, once you, and they might be in tubes instead of bottles, but once you get the liquid, you want to kind of get some of the excess off by just rubbing it on the inside of the tube, kind of pushing on it and wringing it out. And then whatever portion you select, so let's just say I'm going to do behind my ear, kind of rub it real good and get as much as you can. What you're going to do is swab only half of the plate, okay? And then you're going to throw that into the biohazard bag or the bleach, okay? And then you'll notice on page 174 what it says to do is, after you've swabbed your plate, then you take your loop, so you'll flame your loop and let it cool off, and then you're going to go into the original swab that you did and kind of pull a little bit of it out to try to get some isolated colonies, okay? Now we're flaming our loop again. All right. Now I really should flame that because that's my bacteria. Okay, and so once again, your plate you're going to put upside down in the incubator and let it grow, and then the following week you're going to do some different tests on it.